Hello and welcome to the inaugural Dundee PRN vodcast. Today we are talking about neuroanatomy, so take it away.
We'll start with a very basic overview of the lobes of the brain. We're looking at the left side of the brain here with the frontal lobe, parietal lobe, occipital and temporal lobes. If I swivel round, you can now see the cerebellum posteriorly. If we come slightly forwards, there's the spinal cord becoming the medulla as we come up through the foramen magnum. There's the pons, and just rostral to the pons are the two cerebral peduncles. I'm only pointing to the one on the left.
Can we go over the major sulci and the fissures of the brain, please?
Yes, if I rotate the brain so that we can see the superior surface, there's the very obvious central fissure between the left and the right hemispheres. Now let's come back to look at the side of the hemisphere, and you can see quite clearly the lateral fissure separating the temporal lobe from the parietal and frontal lobes above. Now if we stay there, what separates those is the central sulcus that we can see here and running up the lobes onto the superior surface of the brain and then down onto its medial surface, which we can't really see on the model. The reason that central sulcus is so important is that it differentiates between the precentral gyrus here, essentially motor, and the post-central gyrus behind, which is essentially sensory.
Okay, can we now go over the ventricles?
Certainly, Fraser, to do that, let's just take the hemisphere apart, and then we can zoom in on the model onto this blue translucent area.
So this is the lateral ventricle coming back into the occipital lobe and forward into the frontal lobe.
That's right. Now, of course, it's going to curl around into the temporal lobe, but let's show that with a balloon.
So we've got our balloon.
Marge, I just find the ventricles difficult, and I think the balloon makes it easier. So if we think of this whole thing as a lateral ventricle with the frontal lobe pointing towards you, there's an occipital lobe sticking out here, but we haven't got that.
So can we just bring that up here so then we can see what we're trying to show?
Absolutely. The balloon is an elongated version of this.
Okay.
Although we've actually got it the wrong way around. I was talking about that as the frontal lobe, so let's just change. That's now the occipital lobe,
yeah,
and it now matches the brain, and this is extending into the frontal lobe.
Great.
Now what I thought was difficult was how that lateral ventricle became the temporal ventricle, or the ventricle in the temporal lobe, and how structures in the floor of the lateral ventricle end up in the roof of the temporal ventricle.
Mm-hmm.
Yeah? And all it is taking that posterior part and bending it like that, so it now is in the temporal lobe, and what was in the floor a moment ago here is now in the roof of the temporal lobe, and that's the ventricle.
That's it. So has this part got a special name or anything? It's just the horn?
I'm aware of a special name. There is an occipital pole sticking off the back of it, but it's otherwise.
Okay, so what we're saying here is the occipital projection of the ventricle, and then it just carries on going down and bends around.
That's it, Fraser.
Okay. As I mentioned in my lectures, many of the structures develop almost like a ram's horn. So this will curve under, and as it curves under, move outwards with the temporal lobe like that, and ends up very much in that structure.
Okay. And all that we're missing is this occipital
projection.
Occipital projection. Okay. I think that's quite clear.
Yeah.
And so we've got one lateral ventricle here, but there are two lateral ventricles. Where do they join together? Where do they communicate?
Yes, if we take the other cortex, upper portion of the other cortex away now, we can see that there is both a left lateral ventricle and a right lateral ventricle. And the two of them will communicate with the third ventricle, which is buried deep in the center of the brain, just here, through an intervertebral foramen. That's important because the majority of CSF is produced, cerebrospinal fluid is produced within the lateral ventricles, and therefore that fluid has got to flow into the rest of the ventricular system, and it does so by that interventricular foramen, which you would find on either side communicating with the midline third ventricle.
Okay.
How much is it that is produced again per day?
Per day, it's on the order of 500 mils with a volume, total volume of about 125 to 150 mils. So you get roughly 4 changes of cerebrospinal fluid per day.
Okay, and after that it flows back into the venous sinuses via the arachnoid granulation?
Exactly right, principally in the superior sagittal sinus. You should be aware that not all of the CSF is produced in the lateral ventricles. All 4 ventricles contribute, but the majority is produced by the choroid plexus of the lateral ventricles.
Is that just because they're bigger?
I would assume so,
Okay.
yes. Yeah, it's quite an
Simple as
extensive
that.
area of choroid plexus in the lateral ventricles.
And is that the feathery-like area which you can see?
Exactly
in the
right.
ventricles?
It sort of hangs into the ventricle, and it's an extensive vascular plexus, but where the ependymal cells which line the ventricles are reflected back and cover the vasculature and essentially act as the gatekeepers for the material that can flow into the CSF, because the CSF composition is very highly controlled.
So the choroid plexus isn't made of ependymal cells?
It's coated by ependymal cells, but at its core is a vascular plexus. So the blood, there are extensive capillary plexuses
Right, okay.
inside, but there is a coating of ependymal cells on the outside of it, essentially.
Okay.
So your cerebrospinal fluid goes right down your spinal cord up to the end of the sheath around the spinal cord. How does it come back up again to recirculate in the brain?
Yes, good question. I mean, in general, it's just mass flow. There is no real, except of course any movement that you make that moves the cord in any fashion is going to contribute in a sort of stirring fashion, but that's going to be relatively minor. One suspects it's simply mass flow. The odd thing, that, of course, is that the spinal cord does include a central canal down its center. It's quite small, and that is continuous with these chambers. However, it's a blind ending sac, which is rather odd, but it also is full of cerebrospinal fluid, although it's quite a small volume, obviously.
What's the purpose of making so much CSF when we only need 120 mils there? Is it just replenishing it?
No, that's an excellent question. One in which I don't actually know the answer necessarily. I mean, why produce so much of it? One would suspect that the composition is altered with time, as it would be. You've got transmitters being released in the brain, etc., ionic flow in the brain, parenchyma, and so forth, which is going to, over time, change the composition of the CSF were you not replenishing it. So presumably the reason for replenishing it at that rate is to maintain a homeostatic environment in the surrounding of the brain and spinal cord.
But would it be fair to throw the word dynamic in? There are so many things in the body that are just moving and changing all the time on a dynamic nature, and I see the CSF as the same thing. It has to be dynamic to keep it homeostatically correct, otherwise it's just going to become imbalanced very quickly.
I have another question. I saw a surgery the other day. It was removal of part of a herniated disc, and it was down about, I think it was L45 or L5S1, and you could see the cauda equina just like on the side, and it was pulsating. Why was it pulsating? Is that
Pulsating? Well, I mean,
you know, it wasn't like particularly like steady, but it was, you know, there. It was regular enough to see it.
I mean, one possibility, of course, is that each of the roots is accompanied by what's called a radicular artery. So that the roots, if you actually examine them closely, have a blood vessel, or a small artery, which accompanies them up and forms part of the blood supply of the spinal cord, which comes up via these radicular arteries. There's a particularly large one in the lumbar region, but each root will have a radicular artery associated with it. Whether that was part of the pulsation you were seeing, or yes,
That's the only thing I think of. We're close to the aorta. The radicular branches are coming off lumbar arteries very close to the aorta, and I'm assuming that aortic pulsation is reflected in these vessels and therefore causing an apparent pulsation of the cord.
It was really peculiar because I didn't expect. I said, "What's that?" It's the caudate spina. So.
Well, and of course, things like the basilar artery, vertebral arteries are exposed to that subarachnoid space, although they would have been quite a distance away. Whether they would have been contributing or not, I don't know the answer, but potentially they could be. They are running in that subarachnoid space. Whether or not the pulsation of blood flow in them could be contributing is a possibility.
Did you see any veins, Imogen? Because we hear a lot about the internal vertebral venous plexus.
Didn't see any.
No, they could have collapsed, I thought.
Yes, it is really quite diffuse.
During surgery.
Well, to be honest, it just looked like this kind of, I don't know, nondescript mass. That's why I didn't know what it was, and it was just pulsating. It wasn't red. It was just kind of greyish.
You sure the surgeon hadn't gone all the way through and exposed the aorta?
Hope not. So can we go over the main spinal tracts, the spinothalamic, the corticospinal and the dorsal? Because I understand they're the ones that are the most important to know.
Well, the two sensory tracts that we really concentrate on are the dorsal column medial lemniscal system. They're in yellow in this particular model. Someone has colored it yellow, is the dorsal column, and that is carrying fine tactile sensation as well as proprioception, particularly proprioception from the upper limb. That ascends the spinal cord ipsilaterally and only crosses once it has reached the level of the medulla in the brainstem. On the other hand, we have the spinothalamic tract, and that is shown here in blue, and that carries pain and temperature sensation as well as some deep pressure information. The odd thing about the spinothalamic tract, or the different thing about it, is that it actually crosses essentially at every spinal level. So as soon as the information arrives in the spinal cord, it crosses from one side to the other and ascends on the opposite side.
Whereas the dorsal column doesn't do that.
The dorsal column medial lemniscal stays ipsilateral. It stays on the same side that it enters in. It does eventually cross, but it only crosses once it reaches the level of the medulla and synapses.
Why are you saying dorsal column medial meniscal system?
The medial lemniscus is a white matter tract within the brainstem that these fibers eventually join, and that's where that name comes from.
Right.
This is the dorsal column itself. Of course, this is the dorsal spinous process of the vertebrae to orientate you. Now, as far as descending tracts, the main descending tract, the main motor efferent tract, is the corticospinal tract, which comes from the cortex in a single fiber down, and most of the fibers again cross at the level of the medulla and the decussation of the pyramids and end up in the dorsal corticospinal tract, the lateral corticospinal tract that you see just here in orange. So these are fibers which have come from the opposite side and have descended from the opposite cortex. There's a small percentage of fibers that don't cross and that cross at segmental levels, the ventral corticospinal tract, and they're shown just here, but they are very much the minority.
So essentially they cross further up.
These cross at the level of the medulla. The vast majority cross at the level of the medulla, and there's a small percentage that cross segmentally.
Okay.
But clinically, as far as I know, they're not particularly consequential. These are the ones that you have to worry about.
Do you reckon we could visualize this on a skeleton or a model
Yes,
to
that's
see?
a very good idea if we had
Paul,
one.
just before we do that, I'm sorry, can I
please.
come in on the spinothalamic tract again to make sure I've got this right? Am I correct in thinking that